department, and what I want to tell you about is how quantum physics is related to the neuroscience of consciousness. And so I'm going to explain a bit about quantum physics and a bit about neuroscience, but let me start with this word consciousness to say what I mean by that. I'm just a synonym is simply experience. So, excuse me? You can't hear me? I'll try to speak more loudly. So I'm saying what I mean by the word consciousness, and it refers to all sorts of feelings, thoughts, including dreams while we're asleep. All experience is what I mean by this, and so I don't want us to confuse it with fancier ideas like self-consciousness that maybe we think only humans have or something like that. I'm talking about a more basic phenomenon. If you feel pain, then you qualify for this version of what I mean by consciousness. And so in this picture, the mystery of consciousness is represented by the bricks of the pyramid here, representing the physical matter, the physical molecules that we're all made out of, just like rocks and trees and tables and chairs. But some of us have, in addition to the physical matter that we're made out of, it somehow blossoms into these feelings and thoughts and perceptions that we have, namely consciousness. That is a deep mystery that people have been banging their heads against for literally thousands of years now. It's the mind-body problem that you may have heard of. Contemporary philosophers call it the hard problem. David Chalmers coined this term, the hard problem, to refer to this question of if everything's made out of molecules that we don't think have feelings, how do you get feelings when you put them in a certain arrangement in a brain? Okay, so that's the hard problem. That's at the background of everything I'm going to say. It's widely considered insoluble, but by the end of my talk, I want to suggest that we do actually have a conceptual solution in hand right now. Okay, so here's an outline of what I want to talk about in the short time that we have. What I'm going to say towards the end is non-mainstream, and so I want to give you a sense of what the mainstream approach to understanding consciousness in the brain is and what we've learned from that. Then I'll change direction or change tack a bit to come at this problem of what consciousness is from the point of view of trying to understand how certain gases, anesthetic gases, reversibly make us unconscious without breaking us, without killing us. It's a medical miracle that we've been benefiting from for over 100 years, but we still don't understand how those chemicals make us unconscious. So I'll talk about that a bit, and that'll lead us to the idea that microtubule proteins, that I'll tell you more about, in neurons in the brain may be the substrate or the basis of our consciousness in the brain. And then that'll lead me to the idea that it's not only these microtubules in the brain. Thank you. Is that better? So sorry. Please ask me to repeat something if you didn't hear something I said. I said consciousness is experience, and we're going to try and figure out what corresponds to it in the brain. Thank you. Okay, so I'll start with a taste of the neuroscience of consciousness, a bit more of the neuroscience of consciousness from the perspective of how anesthetics work, and then I'll go to this quantum idea, introduce what you need to know about quantum physics to understand what I'm going to suggest. It's all understandable. What I'm going to say today is understandable without fancy mathematics that you might be worried about with quantum physics. Okay, so neuroscience of consciousness. Neuro refers to neurons, which are the brain cells. You know, we've got a bunch of other kinds of brain cells besides neurons in our brain. This is a picture of a brain inside a person's head. There's her eyes. We have other brain cells besides neurons, but we're focusing on the neurons because they are electrically active. They receive signals from our eyes, ears, and the rest of our body, our skin, through nerves that enter the brain. And that, so those are the inputs, sensory inputs to the brain. And then the brain produces motor outputs, we say, to activate muscles, to cause you to behave in any way you can behave, including my speaking to you now, is because neurons are activating muscles in my throat. Okay, so that's what we're gonna try and get an idea what happens in the brain when a person is conscious of something, to start to understand what the basis of consciousness in the brain might be. So you probably maybe have heard that neurons are, or I just told you that neurons are the electrically active brain cells. Here's a schematic of their distinctive shape, where they get inputs from other neurons or from sensory receptors like in our eyes or skin. They get inputs in their dendrites, and then they send outputs throughout their axon branches or axon terminals here. So they have an input side and an output side. Those outputs might go to other neurons or to muscles, again, to cause behavior from that animal. I mentioned they're electrically active. The way they communicate with each other and the way they activate muscles is by, we say, firing action potentials, these pulses, electric pulses of the potential, electric potential across the membrane of these cells. I'm not going to go into depth about this, but again, this is when they are firing spikes. That's another name for these action potentials. These are the signals, the language they use to speak to each other, or again, to activate muscles to control our behavior. And so, based on our interest in figuring out what consciousness in the brain is, we're going to look for what neurons are active in this way, which neurons are firing more action potentials when you're thinking of a particular thing or perceiving a particular thing. And on that basis, we try to figure out what is the neural substrate, or people often refer to the neural correlates of consciousness. What aspects of brain activity, so these are little schematic neurons in this person's brain. These vertical lines are representing the action potentials, the pulses of electricity that are the signals by which they represent things and send outputs to the muscles. And so in searching for these neural correlates, or what I'm calling the neural substrate of consciousness, we're looking for what pattern of brain activity corresponds to a particular perception or thought or idea. And so we record these by putting electrodes into the brains of non-human animals, occasionally even into humans, and try to correlate what they are seeing with what the neural activity is in the brain. Now, I'm not going to go into much depth about this, but one neuroscientist, in order to approach this problem, there's an issue that changing sensory stimulus, like turning the lights on and off or a sound, can cause neural responses, can cause action potentials from neurons in your brain, even if you're in deep sleep. So not every action potential is necessarily going to be related to what you're consciously experiencing at that moment. So there's an issue of trying to disentangle changes in this brain activity that are just driven by the stimulus versus what actually corresponds to what you're experiencing at that moment. And so to try and get around that, neuroscientists look for situations where the stimulus remains the same, the stimulus that you're presenting to the animal or the person, but somehow their conscious perception changes. And here's an example of that, an example of what's called perceptual rivalry. It's known as the Necker cube. If you look at this two-dimensional drawing of a cube, you tend to see it, even though it's two-dimensional, you tend to see it as a three-dimensional cube, but there's two ways you can see it as a cube. You can see it with this corner in the back of the box, or you can see that corner as being on the front, the top. And if you stare at it for a while, you tend to go back and forth between those those perceptions. And so that's an example where the stimulus is not causing changes in neural activity. It's the change in, it should correspond more directly to the change in your consciousness. So here's an example where you can use those glasses. Maybe we can even turn down the lights by the door there. Turn out the lights a bit more. So what you see is a sort of plaid pattern. And if you've got the glasses, the vertical red bars are only going into one eye, and the the horizontal green bars are only going into the other eye. And when your two eyes get two images that are so different that they're not able to fuse them into a three-dimensional image, instead of seeing the plaid pattern, you see it alternate between all vertical or all horizontal. Please pass the glasses around so everyone can see this.
Let's see if it, wait.
So this is an example of your two eyes are getting the same stimulus the whole time, but what you see dramatically changes, even though the stimulus, again, is not changing. So this is a case where we can do this with monkeys or with humans and look for which neurons are turning on and off as they change their percept from one to the other. Okay. Should I wait another 30 seconds for a couple more people to see that?
Everybody need one? Oh, yeah.
Really?
Oh, yeah.
That table needs some glasses. Can you guys pass them?
Here.
Oh, you got it, okay. Sometimes you do see a patchwork, but usually it spreads and you get one or the other. Okay, that's called binocular rivalry because there's competition between what each eye is trying to tell you you're seeing. I'm going to tell you, maybe we can get the backlights go back. Thank you so much. I said we can sometimes record electrically from single neurons. I'm not going to show you results of that kind of recording, although that has been done in binocular rivalry situations like you just experienced. I'm going to show you instead results using electroencephalogram, EEG, which is non-invasive. You don't have to put electrodes into the brain. And this is the oldest way we have been able to measure neural activity in humans. But it still shows us things we can't see by the more glamorous methods like fMRI that you may have heard of, magnetic resonance imaging, because it shows us things happening on very fast time scales, which is what you can't see with that other kind of imaging. So this is an example where she has electrodes at a bunch of spots on her scalp, and we're just measuring electrical activity. And that the electrical activity, the signals that you record at each of those electrodes on the scalp, is reflecting the synchronized activity of many neurons together near that electrode. Okay, so we're not seeing action potentials from individual neurons like I showed you before. This is collective activity of many neurons together. And this is just to show you what the signal looks like and how it can go into these oscillations, these rhythms, brain waves. And we don't need to worry about this so much, but it's just a way of plotting what frequencies are in that brainwave. But here's the result I wanted to show you, schematically represented from this paper from 2009. So they're recording electroencephalogram, EEG, from the scalp while people are experiencing binocular rivalry, like you just did. Instead of the plaid pattern, in this example, one eye was seeing or was being shown the butterfly, another was being shown a picture of these fall leaves. And during that time, people experience, you see the butterfly for a while and then it switches to the leaves and so on. And in the experiment, they're pressing a button whenever their percept changes. And what they find is rhythmic bursts of neural synchronization, relatively high frequency synchronization, represented by this red squiggle, occurring rhythmically and timed to the button presses when the person says, "Now I'm seeing the other picture." And so the interpretation, and this is one study, but supported by other similar studies, is that each moment of our conscious experience is corresponding to this burst of synchronized activity of neurons in different parts of the brain firing their action potentials together. And then when you have another experience, it's a different burst of neural activity. So that's what I got from that first little taste of neuroscience. It's really a simple picture that we end up with, where each moment, what you're experiencing corresponds. So the way we conceptualize this is different neurons respond or fire their action potentials in response to particular sensory features like colors or shapes, or concepts like grandmother or justice. And whatever you're thinking about at that moment is implemented or instantiated by neurons that represent those different features or concepts firing in this synchronized burst. And so at each moment, as you think of different things, different neurons join that assembly or population, that synchronized conscious population. And those seem to be what make up the frames of the movie of our conscious life. Okay, so that's a bit of how people, what the kind of stuff we're learning from trying to understand consciousness from a mainstream neuroscience point of view. Now I'm going to turn to consider certain anesthetics, the volatile anesthetics or gaseous anesthetics. This is so the first amazing, remarkable thing about these gases is that they turn off our consciousness without ruining us, without breaking us. And that has been incredibly important medically for reducing our suffering. But as I said, we don't really know how they work. So this is a chart from a table from a review of a paper trying to talk about people trying to figure out how these anesthetics work. Aside from the medical miraculous nature of them, I think the next miraculous point about them is that they work on everything alive, not just creatures with neurons like us. So that includes invertebrates, worms and flies, single-celled creatures that have no neurons, and even plants. So in us, on this chart, hypnosis here is a code word for unconsciousness, not the state you might associate with suggestibility and so on. Here it's just a jargon word for unconsciousness. So we don't know if these things are conscious. We suspect maybe they're not, but the sense in which these same chemicals work on them is they stop moving and also stop responding in certain ways. Okay, so I said people are trying to figure out how these chemicals work, and so we've got lists of different molecular elements, cellular elements that people think they might target. Ion channels are proteins in the membranes of all cells, but in neurons they are what are responsible for generating the electrical signals I was talking about. So people focus on those. But these gases bind to a whole bunch of different things, and so there's a tendency to think that it's a combination of them that cause the effect of unconsciousness. But I'm going to focus on one particular candidate here, disruption of cytoskeletal elements, specifically microtubules. So I'm going to tell you more about what those are. I wanted to emphasize this quote from one of the early discoverers of anesthetics. What is alive must sense and can be anesthetized. The rest is dead. So he was almost using anesthesia as a definition of life. Okay, I said I'm going to focus on the cytoskeletal microtubules, and I'll tell you why they're called that. But before I focus on the microtubules, I said there's a tendency, because these gases bind to many different molecular targets in biological tissue, there's a tendency to think that it's probably some combination of these many targets that accounts for the unconsciousness. But there's a number of remarkable facts that have been known basically since the beginning that suggest instead that anesthetics may be working primarily on one main target, even though, again, they bind to many things. But for the unconsciousness, the facts I'm about to review suggest they may be primarily operating on one molecular target. Okay, so there's a fair amount on this slide. I'm going to tell you what each part is. Focusing on this panel A here, there's a description of it. It is plotting what's known as the Meyer-Overton correlation for inhalational anesthetics. These are a bunch of different anesthetics, and what this line shows is that how much you need for an effective dose correlates with how well the stuff dissolves in olive oil. And I want to emphasize also that these are very diverse, chemically diverse molecules. Some of them are just an element. And so this relation suggests that rather than the typical way that drugs work by some very specific interaction into some keyhole that they fit into, that this may be a more non-specific physical interaction rather than what we think of as chemical. Okay, that's thing one. On B here, I mentioned that ion channels are one of the things people focus on as a potential target because they participate in making the action potentials. And when you become unconscious, you do reduce the number of action potentials you're firing. What this plot is showing is that, okay, so here are two different inhalational anesthetics. And what the plot is showing is that if you give half of an effective dose of this one and half of an effective dose of this one, they combine approximately linearly to get a full effective dose. And the reason this is remarkable is that these pairs have been chosen because they have very different effects on particular ion channels. So if you think ion channels are the main target, this argues against that. Because again, you know, this one may act strongly on certain ion channels, this one doesn't much at all, but when you combine them, you get a full dose. Even though if it was primarily acting on action potentials, you should only have a half dose if this one doesn't act on action potentials. Okay, so those are a couple points to suggest that maybe, even though these chemicals bind to many things, their effect on consciousness may be from one primary molecular target. And the molecular target I'm going to focus on as a candidate for being that unitary or main target of anesthetics are microtubules. And in this paper, tubulin in the title is referring to the subunits that make up microtubules. So you can read this as essentially talking about microtubules. I'll explain this more in a bit. But we don't need to unpack too much of this, but here's the title. Anesthetic alterations of collective terahertz oscillations in tubulin, I'll unpack that in a second, correlate with clinical potency. As I say, so this is a theoretical modeling study where they did quantum chemical calculations for how a range of different anesthetic and non-anesthetic compounds bind to these bits in the microtubules, and they reported that the binding to microtubules correlates with the clinical potency. So to make that clearer, what I'm saying is they essentially reproduce this Meyer-Overton correlation by assuming microtubules are the main target. And that cannot be said for any other candidate molecular target. So this is a theoretical study that justifies our considering microtubules as possibly the main target of these volatile anesthetics. So let me tell you a little more about microtubules. As the name suggests, they are actually tubes, and I just told you they're made out of tubulin subunits. So the microtubule is a protein, but it's made out of smaller protein subunits. Each of these red and green pairs is one of those tubulin subunits, and they form a tube. Tiny tubes inside all cells, but in neurons they are arranged in particular ways, and they also have many different types of microtubule associated proteins here forming cross bridges amongst the different microtubules. And this is illustrating how they're lined up in dendrites and axons of neurons. They're called part of the cytoskeleton, so cyto is referring to the inside of the cell, and skeleton because they're rigid tubes, and so people thought of them like bones that give the cell its shape. But they have many more functions than just being like bones, is my message. Here's a picture of some green microtubules stained in a non-neuronal cell around the blue nucleus. To support what I just said, that they don't just act as tent poles to hold up the cell shape, they orchestrate and implement the beautiful dance of the chromosomes during mitosis. So here's a real picture of chromosomes after they've been replicated being pulled apart into the two daughter cells. These are all microtubules, and there's a microtubule structure that is also, it's called the microtubule organizing center. It's organizing the whole thing. Moreover, microtubules are what are in the filopodia and cilia of single-celled organisms that allow them to move. So they do a lot more than just act as tent poles or bones for cells. I wanted to also mention that Alzheimer's disease, one of the primary pathological insults that leads to cognitive decline is the breaking up of microtubules, because a certain form of microtubule associated protein called tau protein falls off the microtubules, and so they fall apart. And so there's some support for the idea that these things inside the cells may be related to our cognition, even though we've been focusing on the membrane, the skin of the cells. Okay, here's where my modest contribution comes into the story. This is a paper from last year from my lab, where we did an anesthetic study on rats. And we had this drug called EPO-B that binds to microtubules. And this is, my main experiments are electrical recordings in the brains of rats, which is much more difficult than what we did in this study. In this study, all we were doing was timing how long our rats took to fall unconscious under isoflurane anesthesia, particular one of these anesthetics. And we found that those who had been given the microtubule binding drug, and it's important that it's a particular kind of drug that can get into the brain, whereas others like this don't get into the brain. So a brain penetrant microtubule binding drug caused those animals to be a little bit resistant, or significantly resistant, to the isoflurane anesthesia. And so we inferred, well, here's how it looked in our result. So average lower latency here is time to loss of writing reflex. That means we simply flip them on their back, and if they don't flip themselves back, we count that as that they are unconscious. That's a standard measure. And here you see them just taking significantly longer to fall asleep if they were treated with that EPO-B microtubule binding drug. So we're inferring that the microtubule binding, if this is a microtubule, the microtubule binding drug is binding to that microtubule, getting in the way of the isoflurane anesthesia, and that's why it took them longer. So here's our abstract. I won't read the whole thing, but just to say our results suggest that binding of the anesthetic gas isoflurane to microtubules causes unconsciousness and loss of purposeful behavior in rats, probably us too. That was the result. But here is where we come to the quantum part of the story. This finding is predicted by models that posit consciousness as a property of a quantum physical state of neural microtubules. So I want to introduce a little bit what quantum physics is, how it's different from classical physics. And again, you don't need math to understand the ideas. So actually, the way I wanted to approach this is that the reason people like me think that this is a likely or promising direction to look for an understanding of consciousness is that the quantum model or hypothesis can solve a fundamental problem in the science of consciousness that I argue cannot be solved by a classical model. That problem I'm going to call the binding problem. So here's how it's expressed in our undergraduate neuroscience textbook. Exactly how the parallel streams of sensory data are melded into perception, images, and ideas remains the holy grail of neuroscience. So for example, the blue of that chair might be represented by neurons in one part of my brain, but the shape might be represented by neurons firing in another part of my brain, but somehow I experience it as one unified object. And that goes for the whole scene, which includes many colors, shapes. I experience it as one thing, but it's spread across my brain. The binding problem is how does it get combined or bound together? These different bits of my experience, how do they get combined into the one unified experience that I experience? So calling it the Holy Grail here makes it sound very important, but it's about all that Baer and colleagues had to say in that book. I like this. This is a portrait of his wife by Dali. I don't love Dali. He was not a great guy, but this is suggestive of this binding problem because we're made out of molecules, but we experience ourselves as one whole thing. Here's how William James expressed this problem way back in 1890. He's an American psychologist and philosopher. He said, take a sentence of a dozen words and take 12 men and tell to each one word. Then stand the men in a row or jam them in a bunch and let each think of his word as intently as you will. Nowhere will there be a consciousness of the whole sentence. Okay? The point here is you can replace men with neurons here and the point remains. These guys are touching each other. The neurons are not even touching each other. They are sending little chemicals across to each other. So that's another way of expressing the binding problem. Why should, just because things are happening at the same time nearby each other, why would they be experienced as one thing? The proposal is that that problem can be solved if the substrate of consciousness is a quantum state, or we can think of it as a quantum vibration or resonance of microtubules spread across many different neurons spread across our brain. Why this is possible, or why this is a reasonable proposal, is that quantum physics is irreducibly holistic in a way that classical physics is not. I'm going to explain that a little more now. Okay, but this is the hypothesis. The substrate of consciousness is somehow vibrations of these little tubes in the neurons in our brains, and it's important that it has to span multiple neurons, can't just be inside one neuron, again, because of the binding problem. Neurons that are representing different things I'm experiencing are in different parts of my brain. Okay, so here we come. I'm going to say a bit about quantum physics. We are told that quantum physics is the physics of atoms and molecules. This is supposed to represent an atom. And that's true, but you get the impression, or people tend to think or even say, that it's the physics of small things, that it only applies to small things. But that's not true. It's our theory of everything. If you've heard this, it is the physics of atoms, but atoms make up everything, or more or less everything. And so quantum physics is the physics of everything. So I'm going to tell you how it is different from classical physics. So that's the dichotomy here. Classical, the old kind. Quantum physics, our contemporary theory of matter. Here's the essence of classical physics. Everything in classical physics can be understood as little bits pushing or pulling on only their neighbors. So we say it's deterministic, meaning if you know the state at the beginning and you know how things are changing, then you can predict, in principle, what will happen forever. The local here is equally as important as the deterministic part. This is the word that means everything that happens in a classical model can be reduced, in principle, to just infinitesimal parts pushing on direct neighbors. There's no action at a distance. That's different in quantum physics. So, quantum physics instead describes probabilities of different measurement outcomes with something we call a system's wave function. So on this picture of an atom, the electron cloud here, this is not representing many electrons. The dots are representing, the density of the dots is representing the likelihood of finding this one electron here versus here. It's more likely to be found here than here. That's one way that quantum physics is different from classical physics. But whereas classical physics just has sort of one part, that is the deterministic evolution, how things change over time, will tell you what's going to happen forever. Quantum dynamics have two parts. One is locally deterministic, like in classical physics, but quantum theory only describes probabilities of getting different outcomes when you make a measurement. Okay, and this, what I call the wave function, is a function that's describing probabilities of different things happening. You make a measurement and you get an answer. That's quantum physics. What the theory gives you is the probability of the different measurement results you might get. Before I make those notes, here's a picture of that idea of a wave function here representing a probability amplitude, something like a probability of finding the electrons, say, at any of these different positions. It's more likely to be found there, but it might be found here. But then when you make a measurement, we use the expression the wave function collapses or is reduced because you find it somewhere. And so now your new probability function says it's definitely there and it's not anywhere else. That's called quantum state reduction or wave function collapse when you make a measurement. So because this is happening when you measure something, it seems like this is just your way of representing, I don't know where the thing is. It might be here, it might be there, and then I look and it's in one place, and so I draw a new curve. So it looks like it's about our knowledge. But it's not. It's more than that. I don't have time to prove that to you. But it's more than, we say, epistemic. It's objective. It's not just about our knowledge. It's something happening in the real world. That wave function collapse. Other people have different opinions about this, but I'm telling you. It's non-deterministic, so I mentioned probabilities, so we've only got probabilities. That's the usual thing, the first thing you hear about quantum physics. But for me, the essential thing, the essential difference is the irreducible holism. And that's not obvious from anything that I've told you so far, But it turns out that when a measurement is made like this, you know, something is happening over here. Even though you made your measurement here and found it here, something is happening physically in the other places because the wave function changed. And that happens instantaneously, as far as we can tell. Recent Nobel Prize was for that, for experimental demonstrations of that quantum holism, or quantum non-locality is the jargon term you might hear. It's another word for quantum holism, starting with an H, not as in hole, starting with a W. Okay, not holes that you dig in the ground. Okay, that was a lightning primer on quantum theory. Quantum theory, as I've described it, is incomplete. This was a complaint that Einstein had, and now Penrose, another Nobel winner. It's incomplete because it doesn't tell us what a measurement actually is in the real world. You know, we are physical systems just like the things we're trying to measure, so it's not supposed to rely on a human making the measurement or something. So does it count when it's just your detector that makes the measurement, or when does the measurement happen? That's an issue that's not solved in quantum mechanics. There are different opinions about how to deal with that. The one I subscribe to is what I said earlier, that something really happens in the real world when the wave function collapses. This is known as objective reduction. Reduction, I said, is another word for collapse of the wave function. Objective, saying it really happens in the real world. It's not just some artifact of we learned something new. Objective reduction means collapse is a real physical process, not just a subjective artifact of our learning something new. It's not epistemic. One more comment. If you've heard of Schrodinger's cat, there's lots of confusion around this, but Schrodinger's point was that you can't leave out the measurements. And that is one of the alternative views that I mentioned. Alternative to objective reduction is that these measurement things are ugly and annoying. Let's just leave them out. If you leave them out, you get absurd predictions like a cat that can be half dead and half alive, which we never see. If you're not familiar with this, it's a thought experiment where we've set up this cruel contraption that will randomly emit a radioactive particle that will cause this beaker of poison gas to be broken and kill the cat, but it happens at a random time. And so at a given moment, at a certain moment, we don't know if it has broken yet. And according to the quantum formalism, if you leave out the measurement part, it would say the cat is half dead and half alive, which is not true. That's why you got to keep the measurements. There really is something that corresponds to those measurements. Okay, so finally coming back to neurobiology, I mentioned Roger Penrose, so he's a Nobel Prize winning physicist who has an idea about how this quantum collapse might happen, and he also has an argument about why he thinks it is probably related to consciousness, which is more subtle and technical than the binding argument that I just gave you, so I'll leave that aside. But so he thinks objective reduction is related to consciousness in a certain way. Not in the sense that it takes a human consciousness to cause a measurement. It's absolutely not that. But he has a theory of objective reduction. Stuart Hameroff is an anesthesiologist who read Penrose's book talking about thinking this is related to consciousness, and Hameroff said, I think the substrate of what you're talking about is microtubules in the brain. And so they proposed their somewhat infamous theory called orchestrated objective reduction. So you've heard what objective reduction is. It means the collapse of the wave function is something that really happens. The orchestrated part, so here's their theory. they suggest that a moment of quantum collapse, when some quantum system decides what it's going to be out of the possible things that it might do or be, the collapse of the wave function is when it decides on one concrete outcome: the cat is alive or the cat is dead. Such a collapse event is associated with what we can call proto-consciousness, some tiny bit of consciousness. It's only in living organisms where biological structures have evolved, like microtubules and neurons, to combine the little collapse events, little microscopic collapse events, into macroscopic collapse events that incorporate aspects of what's happening out there that got fed into the brain. That's when we refer to it as consciousness as we experience it, as opposed to proto-consciousness. So the orchestrated objective reduction is saying, in normal unconscious matter, you just have incoherent, lots of little collapses happening all the time. But in a brain, those collapses are orchestrated into a bigger state, more complex state with experiences like ours. This is very controversial, and so I want to show you that there is experimental evidence for this before we run out of time. So they put forward this theory back in the 90s, the mid-90s, and they've just been ridiculed for 30 years or so, because so far when we try to engineer quantum effects in our technological devices, it seems that we need to make things super cold. And so people thought brains are just too hot and neurons are just too big for these delicate quantum effects to be relevant. And there were always theoretical reasons that those were not conclusive arguments, but now we have experimental evidence to show that that was wrong. So here's the start. So the objection was brains are too hot. So this is a paper about quantum effects in microtubules. They're referring to tryptophan in the title. Those are even smaller bits of subunits of the microtubules inside the tubulin subunits. I'm gonna zoom in on the picture there. But the bottom line is their experiments were at room temperature. That's hot in this context. And they're seeing robust quantum behavior from microtubules. And moreover, so I'm not going to go into the technical details of the effect. It's a quantum optical effect called superradiance. But what's really striking is that the effect gets stronger as the microtubule structures get bigger. Contrary to the prejudice that quantum applies mainly to small things. These quantum effects are getting stronger as you combine the subunits into microtubules into structures like centrioles, or big bundles of them in axons of neurons. Okay, so that's evidence in vitro, not in a living neuron, that microtubules can support quantum effects. Anirban Bandyopadhyay has evidence in living neurons related to this. Again, I'm not going to try and unpack everything on this plot, but this is illustrating that he's performing his experiments on living neurons in a dish, and moreover, more than one. And he's seeing that microtubule vibrations that he can measure and stimulate predict and control the spiking activity on the membrane, the action potentials that we were talking about earlier. And so Bandyopadhyay says that trying to understand neurons by only focusing on the membrane is like trying to understand an animal by only studying the skin. We need to remember that there's stuff inside there, and it seems to interact with the membrane voltage that we know governs our behavior, the spikes, the action potentials. And moreover, it spans multiple neurons, which, remember, we needed that to solve the binding problem. So that's in living neurons in a dish. And finally, the last bit of experimental evidence I'll just mention to you is now in living human beings, conscious human beings, experimental indications of non-classical brain functions. It's a way of saying quantum without saying quantum. So I'm not going to read this whole thing, but so they used a regular MRI scanner, as hospitals have, but they set it in unconventional settings to try and isolate entanglement or quantum effect related signals. And they recorded a signal. I'm going to show you the picture before I read that. They recorded a signal that looks like the electrical signal you get from electromyogram, electrical recording across your heart to see how your heart's doing. But with this nuclear magnetic resonance MRI scanner, which should not be showing that kind of signal. It's not even an electrical signal, but it looks just like the heartbeat evoked signal. And they said that based on their settings, this could only be quantum. There's one paper of someone saying they don't believe it, but they don't have an alternative explanation for the signal. So right now, that's where it stands. And moreover, so that's direct biophysical evidence that there's a quantum state in the living human brain. And moreover, it seems to be related to consciousness because one of their subjects fell asleep in the scanner and the fidelity of the signal degraded. And they also saw the fidelity of that signal correlating with working memory performance. So supporting that putative quantum state in the human brain is related to our consciousness and cognitive function. I'm trying to leave a few minutes for questions. So here's what we said. That was the classical neuroscience result, that we fire neurons that correspond to what we're thinking at a given moment. I showed you some experimental evidence supporting that anesthetics work on microtubules to make us unconscious, and further evidence that it might be because microtubules are supporting a quantum vibrational state that is the substrate of our consciousness. And so that's what I described in this last part as the orchestrated objective reduction theory of Penrose and Hameroff. Quantum collapse in conscious creatures is orchestrated to fuse multiple sensory signals into a complex, unified experience. Is that a question?
It is, yeah.
Yes, please.
It's fascinating. And honestly, I don't understand the quantum collapse and the philosophy around quantum physics. It's very complicated. Exactly. But it seems very reminiscent of solid state physics or carbon nanotubes, where you would matches some of the things with electrical impulses, the larger gas, the tighter.
Oh, yeah.
So is that rejected in this, or is that kind of something? Is that a quantum effect?
The kind of effects you're talking about, I think, where the conductance gets less as you make the thing bigger, that happens in.
You know, the bands would get sharper, better defined.
Okay, it's a different effect.
With, sure, yeah.
In any case, there are similar effects demonstrated in microtubules. Yeah, so they may help us to develop solid state materials. Yes, another
Yes,
question.
have you, it's absolutely fascinating, have you encountered any evidence that microtubules can repair themselves once they've spun apart, as you showed earlier?
Yeah, something I didn't mention is that they're dynamic all the time, or in certain situations, they grow and shrink. It's called dynamic instability. They can even treadmill, where they shrink on one end and grow on the other end. So they have a bunch of mysterious behaviors. So I know that they can reorganize themselves, and the microtubule associated proteins reorganize themselves during learning. So on some level, I think I can say yes, but on the other hand, there are certainly insults that they can't recover from, like in Alzheimer's disease. Yes?
Yeah, very, totally intriguing. Never thought of anesthesia in quite the same way. I was intrigued by the graph that showed the potency of the anesthesia enhanced what appeared to be more lipid solubility.
Yeah.
Which suggested to me as a pharmacologist that its effect is mediated by membrane-related events and its ability to solubilize through lipid membranes. And that made me think, how much of this effect is mediated, microtubules or whatnot, is mediated through G protein coupled receptors? Do they play a role in orchestrating the effect of an external stimuli to more of an architectural rearrangement of microtubules?
I don't know of a role of G protein coupled receptors in governing microtubules. There probably are examples of that, but your point about what I called olive oil solubility being related to lipid solubility is right. So these anesthetic gases go into hydrophobic pockets, and you mentioned the membrane, which is a lipid, and so initially people focused on the membrane as possibly a target, but that seems to be ruled out. So now the focus is on lipidy parts of proteins, the hydrophobic, so-called hydrophobic pockets in these proteins. And as I mentioned, they bind to a lot of them, so it's tricky to figure out which are the relevant ones, the functionally relevant ones. You had.
In the experiments you showed, you didn't show us a collapse. You measured, the experiments showed us that they could measure the oscillations of tubules.
Yeah.
If you measure an electron, as you showed us with the wave function, it collapses. So why use the word quantum for this? You're now measuring a physical property of microtubules. And it may very well, though you haven't shown how that actually affects the function that would lead to unconsciousness. It's a good hypothesis. But
so your
why
question-
why the word quantum at all? Because I think that's what people are waving at Penrose, who unfortunately went a little bit off the deep end, and the anesthesiologist you mentioned isn't very highly thought of because he doesn't do any experimental work at all. But the idea of quantum, I think, is maybe getting in the way of doing the science that this field wants to do.
I hear you. And your point about those resonances is a fair one, but the quantum aspect is essential because a classical model cannot address the binding problem, because any larger scale entity in a classical model is absolutely reducible to local parts. And so if we experience a holistic entity, it can have no physical effects if it is classical. If we postulate that there's some classical conscious correlate of a classical system, it can have no physical effects to give a evolutionary advantage because it can in principle be eliminated from the description. That's different from the quantum case where holistic things can have effects and can provide an advantage, like in terms of memory. And so there are quantum algorithms already combining Grover's search algorithm with a network organization to achieve exponential memory capacity, whereas our classical models are linear in the number of memories they can store based on the number of neurons you have. So I haven't gotten to your question about the resonances, but I want to point out that the quantum part is essential because, in my opinion, and this is, in my opinion, it's more than an opinion because it's an incontrovertible fact that larger scale things in classical physics can be reduced and eliminated. Okay, so but your question about the resonances that I called quantum, where maybe I should have been more circumspect. So this superradiance effect is absolutely quantum. There's no two ways about that. And that's in microtubules at room temperature. Now, I take your point that there's no proof that this effect is quantum. So I went slightly beyond what I was justified in saying there, but we have the previous evidence that they can behave quantum mechanically, and it gets more quantum as they get bigger. And now we see something the microtubules are doing is crossing neurons and controlling membrane voltage. So yeah, it doesn't have to be quantum based only on this experiment, but I think the circumstantial evidence supports that.
If there were a collapse, suddenly there wouldn't be any voltage. The two, instead of being a quantitative change in the membrane potential or the axon, the frequency of spikes, the whole thing would just stop. And then start, isn't that anesthesia? Everything just stops.
No.
Okay, well, and you should let other questions.
Thank you. Yes.
The question, this is related to the previous
Sorry.
questions.
Yes.
Thank you for a very fascinating lecture. But I have some of the same problems. If things happen simultaneously as that they would be if the two states in your different sections of the brain are entangled, something has to have prepared that entangled state so that in fact it can collapse together.
Yes.
Now, related to that question, if we were dealing with vibrations, they could be quantum vibrations because everything, as you said before, is quantum. If those vibrations were transmitted at the speed of light, for example, to the other part of the brain, as they could very well be, you wouldn't need to involve this spooky action at a distance. You could say there is some energy, electromagnetic, let's say, that couples these two parts of the system, which could in fact give you the answer that you want because they're coupled anyway, but without requiring this spooky action at the distance of preparing these very complicated entangled states to begin with, that then all of a sudden collapse.
I understand. So we can debate about what the evidence is so far. I think there's evidence of quantum states in the living human brain and in microtubules at room temperature. But your assertion that it would be fine if you could send light speed signals between different parts of the brain, I disagree with. That is not going to solve the binding problem. Because again, if that's how it's working, you can omit the unified consciousness from the description in principle and predict the behavior of that brain for all time based only on what's happening at local neurons. And so there's nothing in the theory, in the physical theory, to correspond to the holistic experience. So I think we have experimental evidence to support what I'm saying, but also there's a very strong theoretical argument that it simply can't be classical. Yes.
Okay, I'm not a science person, so I don't really speak your language. However, this strikes me as being a different iteration of the Cartesian divide, in that even the brain is being viewed as being separate from the brain that circulates throughout the neurology, throughout the whole body, and that the body itself needs to be included in the binding problem, as you say, which, because of the complexity of our bodies interacting with every part of itself and every part of our lived experience, that it necessarily is part of the binding capacity that we have in our, not just our perceptiveness, but in our interactive abilities. So I'm curious, for example, whether or not how you could bring the whole body brain into your research, perhaps by, for example, looking at quadriplegics who have a paralysis that, because we take for granted the intelligence of the body and how it is included and integrated into our capacity for all the things we're talking about. So just a non-science perspective.
Yeah,
Curious.
no, thank you. So the question of how far this neural substrate extends, might it extend to nerves or other parts of tissue that are not in the brain, is separate from the debate about whether it has to be quantum or anything like that. I mean, it could be that all that information that you talked about from the body is just fed to the brain, and then the correlate of your sensations of the body are all happening in the brain. It's conceivable that the nerves in the rest of the brain are part of that, but on the other hand, we have evidence like you can lose an arm and still feel that arm. So that suggests the feeling is not in the arm. It supports that it's in the brain.
I'm not implying it's one or the other, but it is a holistic system that has a much more integrated intelligence than perhaps that's brain model.
Yeah, this viewpoint clarifies the continuity between consciousness and life. That is, I mentioned the same anesthetics work on single cells. They act like dogs and crabs. If you look at them through the microscope, poke them, they're running around, they're sniffing. It might all be just machine. It is some kind of machinery in there, but this same dynamic on the microtubules that I'm saying we have orchestrated into a higher form of intelligence could already been used, apparently is already being used, was already being used by single cells to do their adaptive behaviors. Yeah, I'm not sure if I fully got it, your question. But so the emphasis on a holistic perspective, I appreciate, but there is a sharp distinction between the quantum holism that I think is limited to the brain in this context, as opposed to something that might be spread farther. Yes.
Are you aware of any research being done with the compound FOB on Alzheimer's, for example? Or
No,
other
but
brain-
these drugs, I didn't mention that this class of drugs that EPO-B is in are cancer drugs. So they're widely used. Most of them don't get into the brain, but because microtubules are involved in cell division and cancer is runaway cell division, these drugs are used as anti-cancer drugs. And so I do know that they are being investigated in a bunch of other neurological disorders. And they've proven helpful in some of them, like regenerating spinal injuries, but I don't think, and so that pertains to your question about repairing microtubules as well. But I'm not sure about Alzheimer's. One more, yeah?
What is the underlying energy support for these microtubules? So what does mitochondria got to do with it and respiration? And how do these tubules maintain their level of energy?
Yeah, they are intimately related to mitochondria, which are also affected by the anesthetics we've been talking about. They use GTP instead of ATP for their source of energy to build themselves and also to move themselves. So did I get at your question? Where they get their energy from? It's
So, and if you look at these
Aliens.
acids,
Simple answer.
membranes, they affect mitochondria and they affect indirectly these tubules.
or directly.
Or directly, and the question is, how are they acting in more than one way?
Oh, they absolutely are acting on many targets, and so they've been demonstrated to have effects on ion channels, like I mentioned, and so on. It's just that those, thinking of it as a combination causing the unconsciousness does not account for things like the Meyer-Overton correlation that suggests it's one main target. Thanks all, I'm happy to talk further.